Good afternoon. So I'm Shanmugavel Chinnathambi from National Institute of Material Science. So my presentation title is the Functionalized Near-Infrared Quantum Dots for Biological Applications. So we know about the quantum dots. It's a semiconductor nanocrystals that are very small size, around 2 to 10 nanometer, and 10 to 50 atoms in diameter. The size and shape and number of electrons are precisely controlled. So in my studies, I used two types of quantum dots. So one is cadmium selenide isotenous. This is a core shell type of quantum dots, and another one is silicon quantum dots for drug delivery application. So first, the cadmium selenide is the more toxic. We know all about cadmium, and silicon is biocompatible mostly. The first I made the micelle about cadmium selenide and iron oxide as a core using the phospholipid as a micelle. So this is the preparation method of micelle. After the preparation, I used the magnetic separation. In this purpose, I used iron oxide quantum dots inside the core of the phospholipid micelles. The iron oxide nanoparticle I used around 5 nanometer, and the quantum dot size is around 7 nanometer. So in this case, I used biotin as a free end of the this lipid because of some additional advantage of these studies. So the first advantage is the fluorescence intensity. We want to keep the fluorescence intensity is high. The another one is we need the biocompatible quantum dots to make this micelle. And this is the structure of the prepared micelle. The core contains both iron oxide and CDSC or the silicon quantum dots. This hydrophobic terminated CDSC isotenous and Fe3O4 and both nanoparticles contained inside the core. Both nanoparticles were entrapped in the hydrophobic core of the micelle, and the bottom is the original structure. And this type of micelle is useful for carrying anti-cancer agent, can be targeted to the tumor site and accumulated in the cancer cells by the help of implanted or externally applied magnetic field. And I prepared around 4 sizes of micelle. The average size is around here. The varying to vary the iron oxide nanoparticle inside the core. We prepared around 4 sizes of nanoparticle. For my application, I used the small size micelle. So this is the optical and magnetic characterization of prepared micelle, and the first CDSC isotenous quantum dot is in chloroform is a solvent, and iron oxide also. But in this case of micelle, it's water soluble. This is the emission spectrum of both CDSC isotenous in chloroform, and the red color shows the micelle in water. So there is no difference in the fluorescence emission after making the water soluble. And this vibrating sample magnetometer reading shows the core contain both magnetic and CDSC profile. And also there is one more drawback to make the water soluble quantum dots. So there is some lifetime of fluorescence excited state lifetime is little bit decreased, but it's more stable than original quantum dots in chloroform. So these first TEM images shows the CDSC isotenous core shell quantum dots in chloroform. So it's well dispersed in chloroform, but the problem is it's not water soluble. That's why we are going to make this type of micelle. This micelle contains both this is the EDS profile of this micelle, single micelle. So this figure shows the separation using neodymium-based magnet about 1.21 Tesla. After this separation is not easy. It will take more than 1 hour, 1 or 2 hours. Sometimes it depends upon the size of the micelle. It's very difficult to separate this type of magnetic separation, and also we need some high power magnet. And the next one is we want to check the photo stability compared with original CDSC quantum dot in the chloroform. This first figure shows the CDS ZNS in chloroform after the 265 nanometer UV light irradiation. It's a 60-minute irradiation, almost gone. But in the case of mycel, so after 1 hour irradiation, still it has some good fluorescence intensity. This is the normalized comparison of the both CDS and the mycel. So this is the first one is mycel, and second, this is the after 1 hour it will completely degrade to compare with the original one. And this is a cytotoxicity of my prepared mycel. I used all the 4 sizes of mycel, so almost it's 100% cell viability, both HeLa cells and A549 cells. So after that, I'm going to checking how to this missile interact with the human serum albumin. It's contained in blood. This is the absorption characterization of human serum albumin interaction with mycel. This is the simple method to evaluate the interaction between the small molecule and macromolecules such as protein or DNA. This 260 around 278 nanometer peak comes from the aromatic amino acid presence in the human serum albumin. After serial addition of this mycel, the absorption will increase after addition of the serial concentration of this mycel, and also in this case of polypeptide backbone, also get some difference of after addition of the mycel. So after this experiment, you have to calculate the binding constant, how to interact with this mycel with human serum albumin. So we have to calculate some binding constant. It's the 10 power 4th order. It's a good stability. The next one is steady state spectroscopic studies of this mycel, how to interact with human serum albumin. This is the emission spectrum of this protein. After addition of this serial concentration of the mycel, the fluorescence quenching is happen. So I checked around the four temperature, room temperature to around 40 degrees Celsius. If I increase the temperature, the fluorescence quenching is happen very fast. And also we calculate the quenching constant about this one. So we have 2 types of quenching. One is static and dynamic quenching. So the next slide shows the time resolved spectroscopic studies. There is any changes in the lifetime excited state lifetime. There is some changes, but it's very small. So from the steady state to compare with the steady state and time resolved fluorescence spectroscopic studies, the conclusion is it's a static quenching, and also they have calculated about the enthalpy and entropy value of the different temperature. So most of the main conclusion is the hydrophobic interaction and some hydrogen bond interaction is stabilize this mycel and human serum albumin interaction. And also we have to confirm, shows the secondary structural changes of the protein. And the Stokes shift spectroscopy is very important for secondary structural changes of the protein or main things. This is the this 50 nanometer is the difference between excitation and emission monochromator difference. This is for tyrosine residues, and for tryptophan is around 60 nanometer. 60 nanometer. After interaction with the mycel, there is some quenching is happen usually, but there is some redshift in the case of tyrosine residues, but not in tryptophan residues. This Stokes shift spectroscopy concluded there is some microenvironmental changes around the tyrosine residues. And this is the excitation emission matrix of the same complex. The peak A and peak B. There is 2 peaks present in the human serum albumin. The first peak A is contain polypeptide backbone structure of C11O, and second one is the spectral behavior of tryptophan and tyrosine residues. In the case of absence of mycel, the fluorescence intensity ratio is this one, and after addition of it will a little bit decrease. This means the PK and B revealed that binding of HSA micelle induced some microenvironmental and conformational changes and also the unfolding the peptide structure. This is the circular dichroism study of the same profile. And also from this study, circular dichroism study, so we can calculate the alpha helical content of the protein. So usually the human serum albumin contains around 65 to 80 percentage of, it depends upon the purity of the protein. But in this case, it reduced the secondary structure of this protein. That means alpha helix. This is the formula to calculate the alpha helix content in the protein structure. There is two bands around 208 and 222. These bands are contributed due to the N to pi star transition of the peptide bond of alpha helical structure in protein. This variation is some limitation. If you can limit the how much micelle you can deliver to the drug delivery applications. If you change this a lot, the structure will collapse of the total protein structure. And also fluorescence resonance energy transfer is important for this type of interaction studies. We can calculate between the donor and acceptor distance. But there is some rules, only 3 kind of rules. The donor should have more fluorescence quantum yield, and this overlap area is more, we need more than 30%. And the distance between the donor and acceptance must be less or equal than around 8 nanometer. This is the emission spectrum of the protein, and this is the absorption, the micelle absorption spectrum. This overlap area is around, so all the this overlap area shows the around more than 30%. And finally, we can calculate the distance between donor and acceptor is around 2.37 nanometer. This result concluded there is some energy transfer between the donor emission and acceptor. And also, we checked the prepared micelle after 24 hours and 72 hours in cancer cells, HeLa cells, and lung cancer cells. After 24 hours and 72 hours, we have some good fluorescence. This is after 24 hours in HeLa cells and Fi49 cells. And this slide shows after 72 hours, it will we have some good fluorescence. So next, I found after 72 hours uptake, I collected the cells from the plate and reseed again. After 72 hours, they will get some good fluorescence intensity. That means after cell division, the divided cells also contain some good fluorescence of micelle. That means that prepared micelle is much stronger fluorescence after a long time. And also this is the one more, this is like future studies. Now I'm doing to detect some immunoassay for antigen detections. So it's like ELISA test, but I'm using the secondary antibody as this prepared micelle. The conclusion is this, this is the biocompatible micelle preparation using phospholipid. This fluorescence signal amplification of the quantum dots is much more after the preparation of the micelle to compare with the original one, and the cellular uptake of the micelle is very long time. This kind of the micelle entered in the cancer tissue through blood vessel. It depends upon the size of the micelle. And this second study is I use the silicon quantum dot. The previous one is completely CDSC and isoternus. And second one is I used the silicon quantum dot as a carrier for the CpG ODN. This is a single stranded DNA. In this case, I used pluronic F127 as the surfactant to make the water soluble of hydrogen terminated silicon quantum dots. And after that, we need some positive surface charge to take the negatively charged single stranded DNA. So in this case, I used PI 600 to make the positive charge of these quantum dots. So after that, I can use the production of some cytokines like interleukin 6 and IFN alpha. And this is the TEM images of, usually silicon is biocompatible but not in water soluble. It's really difficult to make water soluble for silicon material. This is the original TEM structure after the preparation. This structure shows the after some hydrofluoric acid etching. Still it's not soluble. After the third image shows the after the hydrolyzation of using some surfactant. I used this surfactant to make water solubilization. So one more option is the silicon is the near infrared fluorescence. Okay, maybe we can use the animal study or something to deep tissue penetration. And this is the character, and this slide shows the characterization of the prepared quantum dot. This is the highly crystalline and soluble in water. The average size is around 5 nanometer, and NIR emission, almost the neutral surface. So this slide shows the PEI modification of the. I think this already I told. These quantum dots have the positively charged PEI, and after that, the negatively charged DNA will uptake the by silicon quantum dots. So this is the CpG-ODN structure I used. This CpG-ODN structure contains a lot of CG sequence frequently. Because the CpG is recognized by toll-like receptor 9, is localized in endolysosome. So I used around the PEA concentration around 0.5, 1, 2, and 3%. After this percentage of variation, it depends upon the DNA uptake. So 0.5% takes around zeta potential values plus 1.7, and then if increasing, it's around going to a plus 8.32. This is the value of after uptake of the CpG-ODN on the surface of silicon quantum dot. And also this fluorescence microscopy confirm the presence of CpG-ODN on the surface of silicon quantum dots. And also these TEM images also support the presence of DNA on the surface of silicon quantum dots. And this slide, this is a fluorescence confocal image microscope. This is just uptake of the 0.5 PEI and 3% PEI. This the measured image clearly shows the uptake is very well. And also I checked the cell viability, 24 hours exposure and 48 hours exposure of the PBMC cell lines. It's very nice. And this is the cytokine production level using this DNA, CpG-ODN DNA on the surface of silicon particles. This axis shows the surface charge of the nanoparticle and the production of interleukin 6 and interleukin alpha. So in the case of interleukin 6, after this around 3% of 3 millivolt, it will almost saturated. But in this case of IFN alpha, it's continuously increasing. It depends upon the surface charge of the nanoparticles. And these confocal images also support the previous graph. And this is the LAMP1 fluorescence from the late endolysosome, and this is the FITC labeled single-stranded CpG-ODN. The measured image clearly shows the this quantum dot is localized in late endosome. It will produce the interleukin 6 at maximum level. Maybe I will. This figure shows this is the after DNA uptake on the surface of silicon quantum dot. If it is interact with TLR9 in early endosome, it will produce it will activate this type of IFN alpha production. And if this type of quantum dot interact with toll-like receptor 9 in late endosome, so it will activate the interleukin 6 production. And this is my conclusion. This observation demonstrates that the balance of bifurcated cytokine induction can be regulated by the surface of the nanoparticle for these studies. Yes. And also, I would like to introduce this field, Professor Nobutaka Hanagata, Director, Nanotechnology Innovation Station, National Institute of Material Science, Japan. And also, he's my guide during my PhD time and the postdoc time now. And also, I wish to thank Nori Hidaya Abu from Malaysia to support these studies. Thank you very much.
Any questions for our speaker? One question is, you use PE for DNA delivery. How do you ensure that your DNA is released, or can you regulate this release from your nanodots? Your DNA, it's basically you use PI for your DNA attachment to your nanodots. So how do you ensure that they are released in the cells? Are they stuck on these nanodots, or can you regulate this release?
How to release?
Yes.
So, we check the different pH.
So it's
values.
pH dependent.
Different pH value. Maybe the late endolysosome have around pH around 5 pH value. We check the release profile in the different pH.
Okay. All right. Thank you.
Yeah.
All right. Thanks once again.
Thank you very much.
Here's your certificate.